about their environment and change their behavioral responses in light of the new information. In other words, there is no central or external control. A very good example of this perhaps is the sand pile. We all have done this, with sand or with sugar or whatever. If you keep on, piling sand at one place, one point, you will see that eventually a mountain will be created and a critical point will reach, and at that point you will see lots of avalanches and the surface will flatten. If you keep on pouring the sand further, then the mountain will again start to build, and at some point a critical point will be reached, and again there will be lots of avalanches. It's a very good example where we see that this is a critical self-organized point, because if the slope is steep, the system tends to have small avalanches, and if it is flat, the sand accumulates. So the system self-controls in a constant critical slope. Again, apart from pouring the sand, there is no other, in fact, in that heap of sand, that sand pile, there is no central or external control. The third, I think perhaps most important hallmark, I think this is the one which defines a complex system. This is probably one which is most important, is emergence. This is the single most important distinguishing feature, emergence. Complex systems produce surprising behavior. In fact, they produce patterns, observations, and properties that just cannot be predicted from knowledge of their past observation. So take, and there are huge numbers of I think the one I have taken here is water molecule. Water molecule, we know, 2 hydrogen, 1 oxygen, H2O, water molecule. On its own, hydrogen and oxygen are, perhaps gaseous, perhaps not useful elements on its own, but they come together, form water, and we all know the power of hydrodynamics. Water can have floods, if there is excessive rain, then you can have a flood situation, that can so on and so forth. You see, erosion is a phenomena that geographers study very closely. But here, studying just one water molecule is not going to explain you the hydrodynamic properties of the erosion or the flood and all that. If you want to understand the flood, then perhaps you need a holistic approach to understand the hydrodynamics of the flow of water. So, in other words, emergent, which is a very important property, is really when the whole is not only more than some of its parts, but can also be very different from its parts. And in fact, emergence arises from the way that the components interact with each other, and not just from their individual properties. And emergence does give rise to nonlinearity and chaotic system behavior, and there are numerous examples of such emergent behaviors. One example is, of course, recessions and booms are emergent behaviors of national economies. Beyond a certain threshold, emergence may even lead to new principles. And it is a one-way traffic, really. The new principles and features may be sometimes detectable from, but not reducible to, those operating at the lower levels of complexity. Now, so those are some of the hallmarks of a complex system that I have talked about. This then swiftly takes me into some observations, in particular both observations inspired by biology and some of the observations where the complex system behavior can be engineered into a system. And I will start with one that you all know. Life, are we an emergent phenomena? We all know that at any given time, some 75 trillion cells are doing what the cells do in the body, but they are constantly dying and being replaced by new ones, such that, as we know, in less than 2 years, we won't have a single cell that we have today. Yet we remain who we are. You remain what you are. How is this possible? Even though each of our cells contains our entire genome, one would hardly say that any one of our cells constitutes us. But taken together, so remember, sum of the parts, the whole is more than the sum of the parts. Taken together, that amounts to what we are, our behavior, our thinking, etc. If we look around us, there are many systems, whether natural or human, that is human engineered, which have complexity or are complex and hard to understand. Probably the most pervasive example is the biological evolution 3.5 billion years ago, the journey from a simple blue-green algae to the emergence of a variety of life forms as a result of reproduction, mutation, natural selection that has required no outside agency to guide it. And so again, you see here adaptation and self-organization. Now, here is one example which I will talk about. In fact, even the example that I've given in the previous slide, the biological evolution, as many of us know and many of us use it, genetic algorithm we use in our computational environment is actually inspired by that theory of evolution, the theory of genetics, and we all use it. Whether we use those algorithms to solve difficult problems in engineering or economics or biology, we all use it. So that's one algorithm we all are familiar with that is heavily used in biology as well. Another observation that of flocking behavior. You see here irreducibility, adaptability, as well as self-organization and emergence. As we know, the beautiful pattern that these birds form, the starlings, as we have seen in some of the birds, when they flock together, so is everything okay? Is everything okay? So there are three simple principles that the birds flocking together use: separation, alignment, and cohesion. Separation is simply steer to avoid crowding local flock mates. Alignment is to do with steering towards the average heading of local flock mates, and cohesion is steer to move toward the average position of local flock mates. You can see there is computation going on in that flocking behavior. If you want to understand flocking behavior, it's no good taking one starling or bird out of that flock. So it's really difficult. Adaptable, these three principles, that's what we use to adapt. We use vision fluid, we use the influence in air current. They self-organize themselves. There is no external control. There is no conductor. There is no queen or king. There is no leader. And as a result of that, what we see is emergence. So here again, this beautiful pattern that emerges is because of this complex system behavior, which you will not see if you take one or two birds out of that flock, but rather you need the whole flock together. So this is now those of us who use heuristics and meta-heuristics computational algorithms know these flocking algorithms we use, the heuristics we develop, the flocking behavior to solve difficult problems. Now here, as we know, I will repeat that these techniques can be used, in engineering or in biology. That's a complicated technique that most bioinformatics research have used, including I have used a number of these inspired techniques, solving biological, bioinformatics problems. Another behavior similar to flocking is swarming. Swarming, here again, the big bang, there are no conductors. The behavior you find in many animal species, from locusts to schools of fish or flocks of birds. What we see is emergent structures commonly, find in various places. The emergent structures are a common strategy on many of such animal groups. Colonies of ants, built by termites, termite castles where we travel on the roadsides, swarms of bees, shoals, schools of fish, flocks of birds, and packs of mammals. Again, there is no blueprint, there are no traffic lights, there are no conductors, there are no leaders, there are no people who, but we see beautiful, self-organized, emergent behavior. To understand again these, we try to understand the holistic system, not by separating them out. Again, the applications of swarm intelligence, numerous in various fields, including biology, where people have applied meta-heuristics to understand biological phenomena or biological problems, solve rather intractable biological problems from computational point of view. So those are some examples where one draws inspiration from nature or from biology, and also applies those into the biological problems, or not just biological problems, but also numerous problems, including commerce and economy. I will then move towards a human engineered system, how complexity can be taken or made use of in a human engineered system. And here is a wonderful example that we all know, nanotechnology, which we know is the study of the control of matter on atomic molecular scale, atomic or molecular scale, 100 atomic scale. Now, this nanotechnology, as we know, so there is a lot of computational work in this. This makes use of or manufacturing material is a new approach which is based on molecular self-assembly, as we know, which is a process by which molecules adopt a defined arrangement without guidance or management from an outside source. So it is a wonderful example of human engineered system where complexity is used to create a valuable material, a very usable material. And there are numerous other examples where complex system phenomenon is found or can be seen. Cyclones, tornadoes, weather systems are emergent features of motion of air, particles on the spinning earth, air pressure, oceans, sun, and carbon dioxide. Now, and there are numerous such examples, and we are seeing as we speak currently the changes in the global weather phenomena, El Nino effect, and all that, excessive rains, and in order to understand these things, you need to see the holistic system as opposed to breaking down those down into constituent parts. All of these systems that I have just mentioned require mathematical modeling, and to prove that the mathematical apparatus is well developed. In fact, with the advent of powerful computational techniques, the mathematical apparatus has developed further. Mathematics has always developed very well. There are huge numbers of, large numbers of excellent mathematical developments and techniques taking place all the time, and computational methods are only enriched mathematical methods. And likewise, mathematics has benefited from computation as well. So, the new mathematical theories have benefited from the development in computational power. In a similar way, there is that synergy between biology and computation. But we live in a very multidisciplinary world, interdisciplinary world rather, not just multidisciplinary. And when we talk about computation and biology, of course, it does bring in the mathematical aspects, engineering aspects, and all the cognitive challenge. As I say, the mathematical apparatus is well developed. The challenge, however, is excellent mathematical modeling, good models, good computation, not just mathematical theoretical models, but also computational models and analysis. And that challenge is in all the disciplines, whether it is engineering or economics or whatever, but equally likely of course, equally in biosciences as well. Now, I've included a few more slides on complex systems. Those of us who come from India, we know the chaotic traffic system in India, which is a very good example of self-organized complex system. No matter how crowded or how chaotic the roads are, and there are no rules, by the way. It's a self-organized system. You will still reach home. And I want to give, in the beginning I mentioned that even simple deterministic systems can exhibit complex system behavior. And here is an example that I want to talk about. We all know a simple pendulum, which Galileo discovered in 1602. It's a simple harmonic motion. We all have studied in our physics textbooks in the schools. There is nothing unusual about that. We all have grandfather's clock at home. You've seen this pendulum, and we can draw this sinusoidal function. So here is a simple harmonic motion that we all understand very well. Now, I don't have, obviously, this is an online presentation, so I can't do the demo for you. But usually at this point, what I do is I demo a double pendulum. So I take two pendulums and I join these pendulums using a rivet that you can see the black part and the yellow part. So I join those, and then I show. So this becomes a double pendulum. So two pendula attached to one another in this fashion. And then I show that this pendulum can show periodic motion in phase, which is predictable. Periodic motion out of phase, also predictable. But if you give a certain motion to this pendulum, then it exhibits a chaotic, unpredictable behavior. I think I have a slide where I can just demo a simulation of this. But this double pendulum is a good example of complex behavior in a simple coupled system. The coupling is coming from that screw which is connecting the black part to the yellow part. That's where the coupling is coming. And it is coupling which brings about that complex system behavior. So modeling is not a problem. One can do it using simple first year mathematics. So you will get certain equations. So on the left hand side of the screen, there is a simple harmonic motion. You will understand that it's predictable behavior. But on the right hand side, you can see more complex behavior or different equations. One of my specialty, of course, is differential equations. Non-linear differential equations, and applying those various problems in science and engineering, including biological problems. So because the work I have done is in non-linear equations and applications in biological problems. For example, the elementary genetics and so on. And, you know, so here these equations are there. You can see divisibility, step quantization. So these are Euler-Lagrange differential equations. Newton's law applies to the double pendulum as well, just like it applies to the. It's deterministic, but it's sensitive to initial conditions. Even if we have exactly the same initial conditions in the double pendulum, we get totally unrepeatable behavior. Now, as I say, it makes more sense when I demo this. I don't know whether you've seen this demo, but it's quite useful to see the demo. And this is the whole area of boundary value problems, initial value problems, non-linear differential equations, and integral equations. And there are numerous applications of these techniques in biological problems as well, including computational solutions. So the double pendulum exhibits an emergent behavior, is a very good example of complex behavior in a simple coupled system. The coupling is coming, as I say, from that screw, that rivet. Each part of the system is relatively simple, easy to understand. Newton's law applies, it's a deterministic system. It is the coupling, new complex behavior, which we understand by now. I don't have, unfortunately, I don't have a demo for you of that. But you know, I will probably keep it for another time. It's an excellent demo to see. I want to move on to one or two more things, finish, and I want to point out some more examples of simple systems. You all know Edward Lorenz, famous mathematician scientist, who was working on predicting weather. He was using a computer once, and he had been used to thinking, and we all think perhaps the same way, that large changes need large forces. But on the contrary, that small changes David Lawrence, in his computer, he was using this strange or Lorentz attractor equations to model the weather behavior. And one fine day, he had to run to a meeting. So what he did was ran the simulation, truncated the parameter using mean 0.50697, truncated that 0.506, thinking that a difference of less than 0.1% is going to make a difference. He ran the off event for the meeting. When he came back, in his own words, this is what he saw. So he says that I wanted to examine some of the solutions in more detail. I had a small computer in my office then, so I typed in some of the intermediate conditions that the truncated value I showed, which the computer had printed out as a new initial conditions to start another computation, and then went out for a while. When I came back, I found that the solution was not the same as the one I had before. The computer was behaving differently. I suspected computer trouble at first, as we all do, but I soon found that the reason was the numbers I had typed, which were not the same as the original one. These were rounded off numbers, and small difference in something became rounded off. We had amplified, in the course of 2 months of simulated weather until the difference was as big as the symbol itself. And to me, this implied that if the real atmosphere behaved in this method, we could not make forecast 2 months ahead. Small errors in observation amplify until it comes. So this is in both words. In other words, when we're doing computation, we see that small truncations can lead to some very abnormal chaotic behavior observations. And one can do this. You don't need a very high level of mathematics here. Here I've given simple simultaneous equations. Paper and pencil exercise will show you that x equal to 1, y equal to 3. Whereas if you try to do this, I think most computers will give you good, accurate solutions these days. But if you do it using a computational model, it depends on what precision value you are using. And the computer solution may be depending on what computer you are using, what is the precision value of that computer, etc. You will get different solutions. For example, the one I have given here in red on the slide. Of course, those were simple simultaneous equations. Can you imagine large numbers of equations, not just simultaneous, perhaps even nonlinear equations, more complicated ones, and the consequences of the rounding off of the errors, the propagation of these errors to the system, and you don't have to go too far in history. 1991, the sinking, collapsing of the oil rig off the coast of Norway, it was found that this was as a result of inaccuracies in the finite element analysis, which is computational averaging process. It's a mathematical technique. So what happened is the value of 1 by 10, which has a non-terminating binary expansion, so that's the binary expansion, was chopped at 24 bits after the radix point, because this is a binary expansion. It is not a decimal point, it is a radix point. The small chopping error multiplied by a large number led to significant error, and hence that collapsing of that oil rig. Likewise, the Ariane 5 rocket in 1996, again not too far, was ultimately the consequences of a simple computational error overflow. So, in other words, small change in the initial conditions produces major and unpredictable qualitative changes. So, it's not necessary that a simple, a complicated complex system will behave at complex system behavior, but even simple deterministic systems can do so. Lorenz's phenomena, he actually, by the way, Lorenz published a very famous paper, very seminal paper in 1972. The paper was entitled "Predictability: Does the flap of a butterfly's wings in Brazil set off a tornado in Texas?" Of course, that does not happen. It's only metaphorical. And since that paper by Lorenz in 1972, this phenomena has come to be known as butterfly effect. But of course, as I say, this is only metaphorical. To mathematicians, this effect is known as sensitive dependency on initial conditions, and it is one of the hallmarks of mathematical chaos, and which results into that emergent behavior we talked about. There are numerous examples. Henri Poincaré, a French mathematician, found an error in a theory, as a result of which this entire new area of fractals has emerged, or the chaotic systems has emerged. Again, there are numerous examples, or rather applications of such in various biological problems, and of course engineering and other problems. I want to give an example of how, as a result of the complex system behavior, an entire discipline has emerged. We all have heard about Mobius strips. Augustus Mobius, 1970, 1868, famous mathematician, who gave this Mobius strip. As a result of this particular phenomenon, an entire branch of mathematics has emerged, which is known as topology. The global structure of space can be strikingly different to its local structure. Now, I don't want to give too many examples. I think I've already given this is another one where we see, you know, there are certain sometimes when we don't have a defined law. So the gravitational law is an inverse square law. We know that, and then you know, there are some other laws. You know, we know those laws. But there are certain situations where we don't have a law, and in those situations, an emergent scaling law emerges. And so these are, you know, sort of again techniques that are used in various applications in engineering science. And very quickly, some of the areas so that is scaling variance or scaling power laws. The complex system behavior. These are important. Understanding of complex system behavior is important to better understand the world and phenomena around us, whether it is stock market, whether natural disasters, avalanches, earthquakes. Apparently, there is some work I have been doing with colleagues in India at IIT, Turkey, where we're looking at avalanches and earthquakes. And of course, these complex system behavior help design useful systems. That's mathematical modeling. But also, how can engineers use complexity into their system? So nanotechnology or composite materials is another example. How do microbes evolve antibiotic resistance so rapidly that we are currently seeing? And that is again a complex system behavior, or can be understood using complex system behavior. Intelligent robots, you know, so an engineering application. My colleagues here at my own institution, my own university, are using behaviors to design robotic applications for medical use as well. How do thoughts, memories, and consciousness emerge from interaction of billions of neurons in our brain? It's an open question. How do different combinations of genes affect a person's likelihood of getting heart disease? What types of interventions are likely to be effective in limiting the spread of pandemics like flu, etc.? With today's computers, we can actually build complete silicon surrogates of these systems inside our computing machines. We can simulate things, you know, and use these would-be worlds as laboratories within which to look at. So that's another benefit of understanding complex systems, the dry labs or simulated environments. Some behaviors, you know, in entertainment, you know, cinema, virtual environments, example using fractals, etc. So that's inspiration used areas like gaming and cinema, etc. Also, of course, most importantly, to develop algorithms by drawing inspiration from natural systems or biological systems and their complex system behavior to solve intractable problems. An example, bio-inspired algorithms, and I've given a few examples already. DNA is a very good example. DNA computing is an inspiration one draws from the way the DNA is structured. So that's DNA computing. It gives you 4 bases to work with as opposed to just two binary numbers. So that's the inspiration. DNA computing is a complete discipline in itself. Uh, and on the other hand, to understand DNA, sequencing, one uses some of these um, algorithms that I have talked about. There are other examples, membrane computing, which is inspired by cell membrane. There's a huge amount of work there. I myself work with my colleagues from Madras Christian College in the area of membrane computing and modeling. And there are so many other examples. Overall, the aim is not to create a complex model, but to create a useful model, a simple model that helps us understand a complex behavior. And then how can we use complexity then in solving some of the intractable, difficult to understand problems? So it's a both-way street. And I've given some open-ended, examples here which could have big impact. Scientists are working on developing self-aware skill using emergent properties of composite materials for a future spacecraft that can sense when and where it has been damaged by a meteorite, for example, and repair itself. Modeling of societal and economic behavior. I've done some work with Professor Arupugam at Krishnagam University in South India on modeling some of the social behavior using some of these techniques. Understanding and managing the huge amounts of complex data, picture it, model it, that's the whole area of data mining or data science. Again, equally applicable to biosciences. And I have some students, as I say, who have finished work. That student I was talking about, who used the power laws, was understanding the phylogenetic or phylogeny, that is relatedness amongst various groups of organisms. And he was particularly using transfer analysis, and that's where he found this long-range correlation amongst those DNA sequences. I precisely don't have results in this particular set of slides to talk about that, but yeah, that paper is available through internet. And then there are others who are working in different areas. Mathematical biology is another area where I am very keen on and interested in. In fact, we do have very high entry at this university. We require a distinction at master's level and a very high level of computational and mathematical skills, very computational skills. So just an open invitation. And I will stop here just with one poem which I'm sure we all have read in our school days, which emphasizes the dependence of events and phenomena upon initial conditions. And this has, as I say, has long been recognized. We all know this poem. For want of a nail, the shoe was lost. For want of a writer was lost, the battle was lost. For want of a battle, the kingdom was lost. It's a beautiful way to describe how small things can have big impact. I think I will end there. Thank you very much for listening to me and for your invitation. And I'm of course more than happy to accept to have any questions you may have briefly. Thank you.
Thank you, sir. Thank you for this informative talk. And now I would request our second presenter, that is from the Hebrew University of Jerusalem, Mr. Liel Sapir, to please give his presentations and keep forward the webinar. Thank you.
Can you hear me now? Hello? Okay. So good afternoon, and thank you for inviting me for this talk. So in our lab, I'm a student of Daniel Harry's lab in the University, and we study the association and stability of macromolecules and we study the stability and structure of macromolecules, and we're specifically interested in the ways to control and manipulate these structures by varying various variables in the solution. So, for example, one way to do this is to vary the, and we know the case of polymers that are temperature sensitive. Another way of changing the solution, the macromolecular properties, is by varying the solution environment, meaning changing the components that compose the solution. So, for example, we can add to a solution that contains macromolecules or colloids, we can add another solute, which we term a cosolute, that affects the structure and stability of the macromolecule or colloid. So in this example, we added these cosolutes, the red spheres, and they affect different structure of the colloid or polymer differently. So effectively, they have a net effect that shifts structural and association equilibriums towards the desired states. And this phenomenon has many manifestations in very different fields. For example, in this slide, you can see that the phenomena of varying cosolutes, adding cosolutes to shift macromolecular association or structure is exploited both in the biological sciences, both in biological systems and in synthetic systems. So on the left-hand side, we have an example of two colloids determined by the shaking group of two colloids. And once they are exposed to a solution that contains an excluded cosolute away from the colloid, they still, so the addition of this excluded cosolute solution drives the association of these colloids with one another. A very different example, but very similar still, is what we can see on the right-hand side. So on the right-hand side, we have an illustration by a good cell of a bacterial cell. Bacterial cell contains many macromolecules of interest, specifically proteins. And the proteins are surrounded by many other such cosolutes that interact differently with the protein. They can stick to the protein, they can stay away from the protein. And this interaction with the surrounding milieu actually in biological environments, many cosolutes sterilize protein in their native states. They shift the protein folding equilibrium towards the native state. And maybe a very interesting example of how cosolutes affect macromolecular or colloidal association and structure is this example of an experiment from the shaking group again, of a suspension of colloids in solution. And these colloids are suspended in an aqueous solution, and to which they added a polymeric cosolute, PEG. And then they checked how this PEG affected the structure of the colloids and how the PEG also affected the temperature response of the colloidal suspension. So what we can see here is that at low temperatures, the colloids associate with one another and form a crystalline structure, kind of a 2D crystalline structure, which they are in the maximum contact they can form with one another. But once temperature, the colloids started dispersing in the solution and form a fully dispersed homogeneous solution. And that's a way of saying that they are in the monomeric state in that specific temperature. When they still increased the temperature further, the colloids started to aggregate again, but this time in another structure, an aggregative state. They started associating with one another. And this means that this research, they were able to change the temperature and shift the colloids from different structures, 3 different structures, depending on the temperature, in the presence of this cosolute. And all of these features in this experiment can be explained by the interaction of the cosolute with the colloid. Specifically, we want to talk about how much the cosolute is sticking or staying away from the colloid. And there are 2 extremes. The cosolutes can either stick to the protein or the colloid or the macromolecule, or they can stay away from it. On the one hand, when they are preferentially excluded from the colloid surface, they actually drive the association of the colloids with one another through depletion forces, what's termed depletion forces. However, when they are preferentially included around the colloid, what happens is that they can shift the structure to specific structure. They will shift it to the structure to which they are preferentially included the most. So in many cases, they stabilize the expanded state. The state in the previous example fully dispersed. But in certain instances that I will talk about in the rest of the talk, they are preferentially included around a more compact state, which we call the compact bridge state, and they act through what is termed bridging attraction to stabilize a structure in which colloids are brought together in space with respect to one another. They are bridged together by a layer of intervening cosolutes. So in a way, the cosolute acts as a glue to stick together 2 colloids. And in the example of the shaken group, one can interpret the results through a shift in the mechanism from a depletion mechanism to a bridging mechanism. So at low temperatures, and you can see it in the respective phase diagram on the right here, at low temperatures, depletion attractions, depletion force acted and drove the association of the colloids into a compact depleted state, in this case a crystalline state. However, when they increase, and that happened when the cosolutes were the colloid. When the researcher increased, the cosolutes began to stay away from began to interact to it, to be preferentially included around it, and thereby they stabilized the expanded state or the monomeric state of the colloids, and the stable state of the suspension was a completely dispersed colloidal suspension. When they increased the temperature still further, the cosolutes stuck harder to the colloids, and thereby they were preferentially included to structures in which they brought together 2 different colloids. So they acted as a glue through the bridging mechanism. So in these beautiful experiments, they could use cosolutes to drive the colloidal suspension between 3 different regimes by varying the temperature. And this phase diagram is by varying the cosolute concentration at a single temperature, meaning looking on the isotherm. And one can also see that there will have an effect on the structure, on this most stable state of the colloids. For example, in this different example of an experiment of polyneprom chain in aqueous methanol solution, they measured the radius of gyration of the polymer in a mixed solution of water and methanol, and they saw that there was a non-monotonic behavior of the radius of gyration with the methanol content. So this is called a re-entrant diagram because it shows this non-monotonic trend. And the radius of gyration begins to decrease at low methanol concentrations, but at a certain point, further increasing the methanol concentration increases the radius of gyration. And the meaning of this is that the researchers mixed 2 good solvents for the polymer, two solvents that cause the polymer to be expanded, but when mixed, they form a bad solvent, a worse solvent that caused the collapse of the polymer is where the radius of gyration went down. So this is called co-non-solvency because it's a very special feature that two good solvents become a worse solvent. And this feature is actually is directly related to the bridging attraction. And I will show later that the bridging attraction has this characteristic manifestation of the size of the colloid or the macromolecule, whereby it starts to decrease at low content of the prelude, but start to increase at a certain concentration. And this certain way, depletion forces and stabilize the depleted state. Bridging attractions induce bridging attractions induced by preferentially included cosolutes drive either the expanded state or a bridge compact state. And all of these phenomena of preferentially interacting cosolutes can be explained through the Gibbs adsorption isotherm. So as you can see in this scheme of the coagulation of colloids in solution, in pure water solution, the because the change in free energy upon the coagulation process is positive. But once you introduce an excluded cosolute, a cosolute that stays away from the colloid surface, it does two things. It destabilizes the expanded state, the monomeric state of the dispersion, but it also destabilizes the aggregative state, the coagulated state. And in fact, it destabilizes the dispersed state to a larger extent, thereby making the free energy difference upon coagulation in the presence of cosolutes more negative. So this means that when the colloids were in water solution, they were fully dispersed, and once you introduce excluded cosolutes, you drive the coagulation. And this can be interpreted in another way. Since the cosolutes are excluded from the colloid surface, water molecules, the solvent molecules, are preferentially included around the colloid surface. And this means that once the colloids associate and form coagulation, water molecules are driven out of the assembly of colloids. The Gibbs adsorption isotherms tells us that the change of the extent of stabilization with cosolute concentrations scales with this change in the number of water molecules that surround the colloids. And this thermodynamically tells us why exactly excluded cosolutes drive the association through depletion forces, and why included cosolutes drive either the bridging attraction or the expanded state stabilization. And what we are interested in our group is to understand what is the molecular mechanism that are preferentially interact with macromolecules, specifically proteins, but also with colloids change the colloid of macromolecule with temperature. And to follow that question, we developed this mean field theory for macromolecules in binary solutions. And we try to describe the following process in which two colloids, the yellow rods, undergo association in a binary mixture that contains both a solvent molecule, the blue spheres, and the green molecules, the green spheres. And to follow the interaction of the two colloids in this binary mixture, we write down the free energy of the system, and we do so by using a regular solution theory called the Flory-Huggins free energy theory, which was originally used to describe protein mixture or melts. And the free energy in the Flory-Huggins solution theory has two major parts. They write it down as a function of the cosolute volume fraction, and it has two main parts: an ideal mixing entropy term and a non-ideal mixing entropy term. And we write down the free energy of the system. We incorporate three adjustable parameters: the size of the cosolutes, which is ni, the size of the green sphere, the cosolutes. Another Flory-Huggins mixing parameter, this is related to meaning the free energetic interaction between cosolute and solvent molecule, and we include an interaction between the cosolute and the colloid, called epsilon, and both chi and epsilon, we count them as free energetic parameters, meaning they have both enthalpic or energetic and entropic components, and we do so because we know that we aim at the temperature response of the colloids or macromolecules in the presence of cosolutes. And since the temperature response depends critically on the enthalpic and entropic components of the free energy, we incorporate these contributions to both these interaction parameters. Chi, the interaction parameter within the solution, solute-solvent, cosolute-solvent interactions, and epsilon, which describes the cosolute-colloid interaction. So this is the basics of the mean field theory, and once we have it and minimize and solve the problem, we can vary the distance between the two colloids in the presence of the binary mixture. And doing so, we can establish this potential of mean force of the interaction of the two colloids in solution. So here you can see the free energy as a function of the intervening space between the two colloids. So when D is very large, complete separation, and then we move them along a straight line towards one another until they are in full contact. And in doing so, we go through the three different at very high separations. We have ended state. I'll call it state E. Once we come to a distance that the two colloids are separated by one layer of cosolutes, we have the bridge state, state B. And once the colloids are in full contact, we have state D, which is the depleted state. And we saw that the depleted state is stabilized by depletion attractions, by region attractions. And now we can vary the various parameters in our model this interaction with between the two colloids. So there is no surprise that when epsilon is very large, meaning there is a net repulsion between the cosolute and colloid, when epsilon is very negative, meaning there are strong interactions between the cosolute and the colloid, meaning the cosolute sticks to the colloid, there is a destabilization of the depleted state, but there is a stabilization of the bridge state relative to the fully expanded state. However, when we increase epsilon, making it repulsive, so the cosolute colloid interaction is unfavorable, we suddenly see in the potential of mean force, or PMF, we see there is a global minimum at full contact. And this means that the depletion interactions, the depletion force acted to stabilize the depleted state, as we can see in this panel here. So we can now change and vary the different parameters and see how they affect the potential of mean force and what is the global minimum of this diagram. For example, we can go to the minimum of the bridge state, and we can look at its dependence here on the y-axis as a function of cosolute concentration. For certain values of epsilon, for example, the black line over here, where the epsilon is very negative, and we said that when epsilon is negative, the cosolute binds the colloid and introduces and drives the region attractions, we can see that there is a net stabilization of the bridge state. However, as we increase the volume fraction of cosolute, this stabilization begins to be weaker and weaker. So there we can, using this model, we can get the reentrant behavior I showed before, and this reentrant behavior is the specific behavior that gives rise to the Condon solvency effect that is known experimentally in a system that contains preferentially included cosolutes. Another interesting thing to do is to take the global minimum of each one of these curves, of each one of these PMF, of each one of these potential of mean force, and see how each cosolute in the knee epsilon space, in the space characterized by the cosolute size and the extent of interaction between cosolute and colloid, which state of the colloid is stabilized? Where's the global minimum? So there is no surprise that for very negative values of epsilon, the bridge interaction will stabilize the bridge state. For very positive values of epsilon, the cosolute will be repelled from the colloid and will introduce depletion interaction to stabilize the depleted state. And for intermediate values of epsilon, the cosolutes will act to stabilize the fully expanded state of the colloid. So by varying these two parameters, in this case, we can stabilize different colloidal structure or macromolecular structure, which was our aim from the beginning, to control the association of colloid or the structures by tuning the properties of the cosolute. And another thing to do is of the cosolute or the temperature. So in this scheme, we show the global minimum, the most stabilized the structure group that will be stabilized by a cosolute in the space described by the solute concentration and epsilon. So you can see that concentration we can shift between regimes. But more interestingly, we can now change the dependence of the colloidal association or the macromolecular process introduced by the cosolute. So for example, let's take a single concentration of cosolute, this white line, and we'll determine the enthalpic and entropic contributions to epsilon. These contributions will directly determine the temperature dependence of epsilon. So in this specific example we show a case whereby epsilon, the energetic component of epsilon and the entropic component of epsilon are both positive. This means that the cosolute interacts with the colloid through a mechanism which is energetically repulsive and entropically attractive. And if we do so, this necessarily means that once one will increase the temperature, epsilon will become smaller. So if we will start at positive epsilon, epsilon will go slower, tend to 0, and then become negative and more negative. And this trend of epsilon explains perfectly the experiments I began with by the shaking group. Because if we begin with a positive epsilon, we know that the depletion attraction will stabilize a depleted state. As we can see here, this is the semi-crystalline state of the colloids in the solution. It was stabilized by action. It stabilized the depleted compact state. And the depletion attracts the most compact state of the colloidal suspension. Once they increased the temperature, epsilon became smaller and smaller until a regime at which it stabilized the expanded state, the dispersed state of colloids, as we can see here. When they increased the temperature still further, they stabilized the aggregative state. And this state was stabilized by the bridging attraction that brought together the different colloids by the cosolute being in the intervening space. So this explains this temperature dependence introduced by the cosolutes. So to conclude, I've shown that the simple mean field theory can account for the effect cosolutes, both preferentially included and preferentially excluded, have on macromolecules or colloids. Moreover, I've shown that including enthalpic and entropic components to these interactions determine the effect of these excluded cosolutes or included cosolutes on the temperature response of the polymer or colloidal suspension. And basically what we've shown is that when epsilon is positive, the cosolute and the colloid are repelled from one another. Depletion forces act to stabilize the most compact state. But when epsilon is negative or negative enough, preferential inclusion will drive either the stabilization of the expanded state of the colloid or will introduce bridging attraction and stabilize the bridge state. I have shown that the entropic and enthalpic contributions to epsilon determine the direction at which this mechanism will tend to, depending on the temperature. And with that, I would like to thank my advisor, Daniel Harris, and the organizer for giving me this chance to introduce my work, and of course all of you for listening. Questions?
Thank you, Liel. Thank you for this informative presentation of yours and discussing your work with us. With this, we come to an end of this webinar. Students from Department of Biological Sciences as well as from the Department of CBBI have gained a lot from this webinar, and I hope that we will carry forward for more webinars in future. Thank you all, and with this I adjourn this webinar.
Thank you.